Welcome to the course on basic genetics. As I said earlier that 3 teachers are involved in this course, Dr. Richa R.A., Professor Madhu Gavaldas Tapadia and myself Jagat Kumar Roy. We are or were from Department of Zoology, Banaras Hindu University. This is week 11, lecture 3. The topic is human genetic disorders, chromosome translocations. In this, I will try to give you some example of chromosome translocation and its mechanism for cancer. We did say earlier that a reciprocal translocation in chronic myelogenous leukemia, that is translocation 9 and 22. This once we have talked. The same thing I am taking in detail, that this is a reciprocal translocation. Here you have chromosome, this is chromosome 9, the longer one, and chromosome 22 is the shorter one. Here you can see very clearly this is chromosome 9 and this is chromosome 22. One part of both the chromosomes are broken, such kind of break can occur. After break, they can get joined also, because in cell always the repair mechanism is very strong. That is, immediately after a part of DNA is broken, joining of that will take place. But while joining, this is possible that the same piece get joined with the same chromosome, or by chance a piece of this comes to the here and the other piece goes there, and they get joined in such a way that this chromosome has part of the other chromosome also. The other chromosome also has a part of other chromosome. The question one may ask, that are there such kind of translocation very common? May not be very common, but definitely there are examples like 922 translocation with chronic myelogenous leukemia and acute lymphocytic leukemia has been commonly seen. So, as I said that this is chromosome 9, this is chromosome 22. Chromosome 22's larger part is going to chromosome 9, but chromosome 9's very small part is coming to chromosome 22. So, result is chromosome 22 has become now smaller. You call it as 22q minus, because the q arm was longer, now q arm is shorter. So, 22q minus. Chromosome 9 became longer than earlier because it has got larger part of chromosome 22. So, you call it as 9q plus, because the q arm is longer now. Very important thing happened with this. I first tell you the genes, that there is a gene ABL that is present on chromosome 9 and there is a gene BCR that is present on chromosome 22. With the result of this translocation, this BCR and ABL, they came together. This is one of the very important event. One may ask question, why precisely BCR and ABL came together? I am not saying always this happens. If this happens, then this can lead to cancer. This is number 1. Number 2, in chromosomes there are break points. That means there are points which can be easily broken. So, in those places the chromosome usually break, and thus these kind of translocation became to some extent common than otherwise it would have been. It is not that any part of the chromosome will get broken and any part will get joined. That kind of example is rarer than the breakage at the breakpoint and getting joined, that is more frequent. So, I will tell you the mechanism, what happens. These are the different regions of ABL gene or ABL protein, I will say ABL protein. This has modulatory region. The modulatory region are Sark homology domain 2 or SH2, Sark homology domain 3 or SH3. The mutation in these can result in the transformation activity or the cancerous activity. This region is the catalytic region. What ABL does? ABL is a tyrosine kinase. So, this is the catalytic region where tyrosine kinase activity is there. Here there is one point that this region is phosphorylated. So, when this region is phosphorylated, its catalytic activity is enhanced. It has suppressor region also, where a phosphorylation makes its activity suppressed. That means, on one side this has modulatory region, other side this has suppressor region. So, this is very well regulated. Now, what has happened? There is a breakpoint. So, this is chromosome 22 BCR gene. There are several breakpoints here. In fact, BCR name has come from breakpoint cluster region. This region had several breakpoints, and that is why this is known as breakpoint cluster region. Earlier this was not known as gene. Later this was found out that it is not only breakpoint those are clustered in this region, but this also codes for a protein, and the name of the protein is BCR. Same thing in c-ABL, that is in ABL gene, there is breakpoint. So, this is a breakpoint here. This breakpoint corresponds to modulatory, a breakpoint in the modulatory region. That means, the modulatory region is now having less regulation or no regulation of the ABL gene activity. So, for CML, this region is broken and this gets joined with this. So, this region plus this region, this is getting joined. So, a fusion protein of 85 kilodalton is formed. In the acute lymphocytic leukemia, the breakpoint is here. So, a large part of BCR and the same region of ABL, they are joined together, and the total protein is 210 kilodalton fusion protein. In both the cases, it is the activity of ABL is now enhanced, that is the tyrosine kinase activity got deregulated, and more tyrosine kinase activity has been related with more cell division. Same thing occurs here, that lot of cell division will occur if this translocation is there. So, this is CML or ALL. Normally, the blood looks like this, where you have so many of RBCs. This is, if I compare it, this is 10 to the power 5 versus this is 10 to the power 3, the WBCs. These are the WBCs which are larger and which are there. And there are very small cells called platelets. So, these three kind of cells are there. in normal blood. But in CML or ALL, you can see the WBC number. Here the WBC number are fewer. Here the WBC number is increased so many. So this is the cancerous condition. In blood, if suppose the WBC number is more, why it is cancerous? Because RBC number is reduced. Within the same region or within the same unit space, you have now more WBCs. And these WBCs are not so much functional WBCs. These WBCs are rapidly dividing, not properly differentiated. So you have WBC number, undifferentiated WBC number increased and RBC number decreased. So this is one kind of cancer we talked about. There is another example, that is Burkitt's lymphoma. Burkitt's lymphoma is also an example of a translocation. This is 8-14, chromosome 8 and chromosome 14 translocation. Let us see what is there in chromosome 8 and what is there in chromosome 14. Chromosome 8 has c-MYC gene, a proto-oncogene. c-MYC is a proto-oncogene. This is associated with cell division. This is this codes for one of the transcription factors. In one of the lecture, I showed that a cell has a relay of the various proteins which signals one after another. Suppose there is growth factor that binds with the growth factor receptor, then you have intracellular signaling one after another. Finally, the transcription factor is in the end, and that gets activated and that enters in the nucleus, allows the transcription of other genes. So c-MYC is one of the transcription factor which enters in the nucleus and allows the transcription of other genes which are required for cell division. Normally this is well regulated, but if it is not regulated, if let us say there is no signal, everything is clean and cell has to keep silent without division. If let us say c-MYC has a mutation which is a transcription factor, without any signal this will start entering in the nucleus and activating the genes which are required for cell division, then that will result in cancer. Same thing occurs in Burkitt's lymphoma. Here you have c-MYC gene that is on chromosome 8. On chromosome 14, you have a gene that codes for IgH, that is immunoglobulin heavy chain. And in the same chromosome, in another locus, there is TCR, that is T cell receptor gene that is present. So in this translocation, if the translocation of c-MYC occurs in that region where IgH is there, so IgH in this, due to translocation on chromosome 14, now IgH is there and c-MYC has reached there. So result is that IgH, this is immunoglobulin heavy chain gene, in immune cell they have to be very highly active. So this is IgH, this is in B cells where immunoglobulin heavy chain genes are active. And if c-MYC reaches there, the result is those B cells, they are proliferated extraordinarily. And thus this is B cell lymphoma. In the other translocation, this happens in the T cells where c-MYC is near to T cell receptor gene, and this is also one of the active gene in T cells, because T cell receptor has to be, gene has to be active, a receptor should be there on the T cell surface, and thus this recognizes the antigen and stimulates the B cell for antibody production. One may ask question, why the translocation occurred of c-MYC with TCR in the T cells and c-MYC with IgH in B cells? It is not designed. If this happens, the thing is, if this happens, if this has come with this, result is, and this is in B cells, result is there will be B cell lymphoma. If c-MYC is with TCR in T cell, result will be T cell lymphoma. So that is what happens in Burkitt's lymphoma. You can see lot of the B cells or lot of the lymphocytes that is there, that is overcrowding, and the other blood cells are fewer. So this is one of the very aggressive kind of lymphoma. So these are the two examples we cited of the translocation. I'll take now the discussion questions. So the discussion points, how common are these translocations? The answer to this is, they are not at all common. Are they well designed or occurring randomly? They can always occur randomly, because I said that there are chromosome breakages that can occur due to high energy radiation or due to some chemical exposure in a cell. We go for X-ray diagnosis, we go for several kind of scans where high energy radiations are used, and the cells exposed, in some of the cells the DNA is broken and getting repaired and so on. The thing is that we have P53 like molecule which looks whether the DNA remains damaged or repaired absolutely okay. So if the repair is not normal, then this situation can become towards the cancer site. So in this case, these translocations, the DNA damage occurred, that means chromosome breakage occurred, and during the process of repair, such kind of translocations occurred. If it has occurred, then this can result in this kind of a problem. The next question is, can these translocations be corrected? The answer is no. They are in every cell. They cannot be corrected. They cannot be taken out and they cannot be corrected. So this is out of question. Are there any kind of treatment that is available? That is the next question. For the CML, that is chronic myeloid leukemia or chronic myelogenous leukemia, there is medicine available, because this is ABL BCR gene fusion. If suppose an individual is heterozygous, in one copy of ABL is normal, BCR is normal, one copy has ABL BCR fusion. The cancer is because of fusion gene product. There are drugs like imatinib and several variants of this. This inhibits the ABL BCR fusion gene product, and thus a relief from cancer is obtained. For Burkitt's lymphoma, I said the cancer is aggressive and the cure is yet to come. It has been seen in both the cases. In Burkitt's lymphoma, in one case the MYC gene has gone to immunoglobulin heavy chain, in other case it has gone to T cell receptor gene. Both these cases there is allelic exclusion, that means one copy is functional, the other copy will have the other rearrangement and will not be functional. So, if one type of IgH is produced in the cell, there cannot be a second kind of IgH produced. So, the second IgH with the same kind of similar kind of rearrangement is inhibited. It has been seen that even if c-MYC has gone to the inactivated IgH, there also Burkitt's lymphoma occurs. This is called position effect variegation. That is, c-MYC, when it has gone to IgH, in IgH gene, there are many factors that makes it activated. There are certain factors which can keep it silent. So, c-MYC gene has gone to IgH, whether it is the allelic excluded or it is the normal one, it is the active one. c-MYC gene has reached there and it is having the factors, those can make it active, and so c-MYC is functional. So, the thing is that Burkitt's lymphoma, or for most of the cancer, the medicine, the cure has to come at this moment. It is not so much there. The only thing is, at early stage, if detected, then one can get rid of. Thank you.